Hello, everyone. I'm Iman Omer, and like Dr. Ellenbogen said, I work in the grid lab for the past 8 weeks, and I'll be talking to you guys about the use of intracranial electroencephalography, or EEG, in understanding methods of anesthesia. So surprisingly, even though anesthesia has been used since the 1800s, the way it actually works to put a person from a state of consciousness to unconsciousness is widely unknown. So in order to learn more, I have analyzed intracranial EEG data using electrodes that have been surgically implanted into patients' brains. And specifically, I was looking at the induction side of anesthesia. So the specific type of anesthesia that I was focusing on is propofol, which is a very widely used type of anesthetic. And at very high doses, an interesting phenomenon called burst suppression occurs, which is characterized by intermittent bursts of activity followed by other intermittent bursts of silence. So looking at the graph on the right, which is a previously collected EEG study, looking at someone's frontal lobe, we can see that underneath the red line marked burst suppression around minutes 8 through 16, we see strobes of blue followed by strobes of red, which in an EEG symbolizes strobes of activity, defined by the red, followed by strobes of suppression, followed by the blue. And this phenomenon is only present under certain types of anesthesia. So this is an important marker when looking at someone's EEG as they're going underneath anesthesia. So looking at our methods, we collected data from patients with epilepsy who were undergoing surgery to have electrodes implanted into their skulls in order to help them with their epilepsy. And I wanted to point out that under Dr. Jay Hoffman and his patients at Seattle Children's Hospital, we were able to collect this data. So shout out to him. And with permission from these patients, we were able to record their EEG data as they were coming in and out of anesthesia. I was looking at a certain patient who was going under anesthesia, and this is their EEG placement plan. We can see like where the different electrodes were being placed. And once we collected this data, I filtered and epoched it. I filtered using a bandpass filter in order to only look at the alpha waves. And after this, I was able to fully analyze the data. So looking at the results, we can see on the left that we can see burst suppression occurring. We can see moments of activity followed by moments of silence. And even though I applied a bandpass filter and we're only looking at alpha waves, this was also present in the raw data. And this burst suppression was present in the majority of the electrodes that I looked at, which is what we expected from previous literature. But if we look on the right at the same time period collected from the electrode on the left, we can see that there is very high activity when other parts of the brain are typically silent. And if we look at where these electrodes that have this atypical activity were placed, we can see highlighted in yellow, they were placed in the anterior and posterior of the insular cortex. So what is the insular cortex? If we look underneath the temporal and the frontal parietal lobe, we can see highlighted in green that this is where the insular cortex sits. And surprisingly, this is one of the least well-known parts of the brain. So seeing that a very unknown part of the brain also may play a role in the use of going underneath anesthesia opens the door to possibilities about learning more about the way your brain is able to shift between consciousness and unconsciousness. So although no definite conclusions can be made, the results do suggest that the insular cortex plays an important role in the shift from consciousness to unconsciousness. And looking at future steps, because I was only able to look at one patient, it's important to look for the future when we do collect more patients, compare to see if these results are also present in their findings, and then also focusing on the insular cortex may help us learn more about not only the way your brain works underneath anesthesia, but also maybe about this unknown part of the brain. And I want to say a big thank you to Dr. Ellenbogen, Mrs. Ellenbogen, my PI and lab mentor, Dr. Heron, and everyone that I've worked with throughout these past 8 weeks, because none of this would have been possible without you guys. Thank you.